Hello everyone, welcome back to Bioenigma Uncovered. I am Chahat, and in today's video we are diving into one of the most powerful topic of bioinformatics, that is BLAST. First up, let's learn how to access BLAST. It stands for Basic Local Alignment Search Tool and it's hosted by NCBI, the National Center of Biotechnology Information. Open your browser and go to BLAST homepage. The link of BLAST is mentioned in the description of this video. Once you are on the BLAST homepage, you will see several options for different types of BLAST searches, most commonly used for nucleotides and proteins. In this video, we will focus on nucleotide BLAST. Use this to compare nucleotide sequences against a nucleotide database. Clicking on the nucleotide BLAST, here you will see a form where you can input your sequence or choose a file from your computer. In the entry query sequence box, you can paste your nucleotide sequence directly if you have a file, or just select choose file and upload it. You need to select the database you want to search against. The default is usually NR by NT, which is non-redundant nucleotide database. If you are looking for something specific, you can choose. Feature allows you to specify which organism you want to search in your search results to come. You can choose to include specific organisms by adding them to your search or remove certain organisms to narrow down your results. You will see an option for program section. Here you can choose how to optimize your search based on the type of sequence similarity you are interested in. Optimize for highly similar sequences, MegaBlast. If your query sequence is very similar to your target sequences, MegaBlast is the way to go. It is designed for fast searches with high sensitivity when dealing with highly similar sequences. This is great for tasks like comparing closely related species or finding nearly identical species. Next, we have Optimize for more dissimilarities sequences. You can choose another database from the list. Contiguous megablast. This contiguous megablast is tailored for searching more distantly related sequences. It is optimized for finding less similar sequences by allowing for gaps and mismatches. This is where the sequences might be more divergent. Moving on to optimize for somewhat similar sequences, that is BLAST N. This is ideal for cross-species comparison. BLAST N option is versatile choice for sequences that are somewhat similar. It is general purpose tool that works well for wide range of sequence similarities and is often used for standard nucleotide sequence comparison. Now let's move to algorithm parameter section. Here we have it. Here you can adjust the settings to fine tune your search. For example, you can change the E-value threshold to set how strict your match needs to be. Once you are set everything up, hit the BLAST button. Your search may take a few minutes depending on the size of your query of the database. If the search is complete, you will see results page with several options. The top part shows a summary of your search and below you will find detailed alignments and graphical views of your results. First is the job title that shows the first query sequence ID by default or more better search identification. Customize it before submission. Then is RID shows the assigned unique identifier to this search for sharing and problem reporting. Download all shows the list of options to save complete search result in desired format such as XML, ASN1. Some list the search conducted, database name, the target database searched. Here is the NT. Next, that is for nucleotide. Next is the query ID. It is query sequence ID for which the result is shown. If a session number is used, it is linked to record the NCBI database. As I use the accession number of the DNA repair protein, that is XPA protein, which is used in nucleotide excision repair. So it is the ID of that XPA protein. Next is the description, is the title of the query sequence from its FASTA defline, that is Homo sapiens XPA DNA damage recognition and repair protein. Next is the molecule type. It shows the type of the sequence, that is the nucleic acid sequence I have taken. Next is the query length, is the length of the current query. One option called as other reports. It links to other reports formats not integrated to the new tab-based display. Now in the graphic, in the description part, here are some other options shown. First is the description. It provides a brief overview of the sequences. Next is the scientific name, shows the organism from which the sequence is derived. Next is the maximum score, indicates the highest alignment score between your query and the sequence. Next is the total score, displays the cumulative scores for the alignment. Query cover shows the percentage of the query sequence that aligns with the database sequence. Next is E-value, represents the statistical significance of the alignment. Lower value indicates higher significance. Next is percentage identity, the percentage of identical matches between the query and the database sequence. Accession length, the length of aligned region and the accession, the unique identifier for the database sequences. That's it for today's overview of BLAST. If you found this video helpful, be sure to like and subscribe for more tips and tutorials. If you have any questions or need further details, drop them in comments below. Thanks for watching and I'll see you in our next video.